In the last HPLC discussion video, we learned about what HPLC is, how HPLC works, and the detection process. In this video, we will discuss the workflow and applications of HPLC in clinical diagnostics. So let's get started with today's video. HPLC has become an indispensable tool in the field of clinical diagnostics. It enables accurate detection and quantification of various biomolecules and biomarkers associated with specific diseases and conditions. The versatility and sensitivity of HPLC make it particularly valuable for analyzing complex biological samples, such as blood, urine, or tissue extracts. There are a lot of applications, which include diagnosing endocrine disorders, as HPLC can measure hormone levels, helping to diagnose conditions like diabetes, thyroid disorders, and adrenal insufficiencies. Detecting infectious agents, as HPLC can identify and quantify viral and bacterial proteins or nucleic acids, aiding in the diagnosis of various infections. Analyzing cancer biomarkers. HPLC can detect the presence of tumor markers like prostate-specific antigen PSA or carcinoembryonic antigen CEA, which can indicate the presence of cancer or monitor its progression. Identifying metabolic disorders. HPLC can detect abnormalities in amino acid, organic acid, and fatty acid metabolism, which are indicative of metabolic disorders such as phenylketonuria or maple syrup urine disease. An example of using high-performance liquid chromatography, or HPLC, in metabolic profiling involves the detection and quantification of hemoglobin A1C, or HbA1c, also known as glycated hemoglobin. HbA1c is a variant of hemoglobin, a protein found in red blood cells responsible for oxygen transport, to which a glucose molecule has attached itself. This glycation is a direct result of persistent elevated blood glucose levels. The HbA1c level in the blood is an indicator of a person's average blood glucose levels over the past 2 to 3 months. It is a crucial biomarker used for monitoring and diagnosing diabetes, as it provides a more stable and long-term measure of blood glucose control compared to single-point blood glucose measurements. Higher HbA1c levels indicate poorer glycemic control, which can increase the risk of diabetes-related complications. Using HPLC to detect HbA1c involves several steps. First is sample preparation. HbA1c testing typically requires a whole blood sample collected in an EDTA-containing tube to prevent clotting. The blood sample is mixed gently and centrifuged to obtain a clear supernatant containing hemoglobin. In some cases, a pre-treatment step such as hemolysis may be needed to release hemoglobin from red blood cells. The sample is then filtered through a syringe filter or centrifugal filter unit to remove any particulate matter and prepare it for injection. Next is HbA1c separation. In an HPLC setup, a cation exchange column, usually used as the stationary phase, differentiates hemoglobin fractions based on their surface charges. The mobile phase typically uses a pH and ionic strength gradient buffer system to control hemoglobin fraction elution, incrementally increasing ionic strength for optimal separation. Post-injection, the sample's hemoglobin fractions, including HbA1c and HbA0, are separated by their ionic interactions with the stationary phase. Non-glycosylated hemoglobin cannot be eluted easily from analytical column because it is more positively charged when compared to glycohemoglobin molecules. Glycosylated hemoglobin can be easily eluted from analytical column because it is little positive charge. Next is detection. As the separated hemoglobin fractions elute from the column, they are detected using a UV visible detector, typically set at a wavelength of 415 nanometers. Hemoglobin absorbs light at this wavelength, allowing for the quantification of each fraction as they pass through the detector. The detector generates a chromatogram with distinct peaks representing different hemoglobin fractions, including HbA1c. Next is data analysis. The generated chromatogram is analyzed to identify and quantify HbA1c. The retention time and peak area of each hemoglobin fraction are compared to those of known standards, allowing for the accurate identification and quantification of HbA1c. The percentage of HbA1c in the sample is then calculated by dividing the area of HbA1c peak by the total area of all hemoglobin peaks. Several manufacturers have developed HPLC-based systems specifically designed for this critical diagnostic test. For example, Biorad Laboratories offers HPLC systems and kits that are tailored for HbA1c testing, such as the D100 system, the Variant 2 Turbo hemoglobin testing systems. Other companies such as Thermo Fisher Scientific, Agilent Technologies, Waters Corporation, Perkin-Elmer, and Shimadzu offer flexible HPLC platforms that can be used for HbA1c testing among a host of other clinical applications. These products on the market reflect the significant role of HPLC in clinical diagnostics, and in particular, the management of diabetes. Also, it is important to note that HPLC, often paired with mass spectrometry or LCMS, offers a powerful method for isolating and measuring proteins and peptides that may act as cancer biomarkers. For instance, prostate-specific antigen or PSA, a biomarker of prostate cancer, and CA125 associated with ovarian cancer can both be identified and quantified using this combined approach. HPLC, when partnered with mass spectrometry, can also identify specific alterations to DNA, such as adducts formed in response to exposure to carcinogens. Therefore, the use of HPLC, especially when combined with mass spectrometry, is a versatile tool in the detection of cancer biomarkers. HPLC offers several advantages in clinical diagnostics. Some of its key characteristics are high resolution, as HPLC can separate components with similar chemical properties, allowing for accurate identification and quantification. Sensitivity. HPLC is capable of detecting trace amounts of components in complex biological samples, ensuring early detection of diseases and conditions. Speed. HPLC offers rapid analysis times, with many separations completed within minutes, making it suitable for high-throughput clinical settings. Versatility. HPLC can be adapted to analyze a wide range of compounds, from small molecules to large proteins and nucleic acids, by selecting appropriate stationary phases, mobile phases, and detectors. Despite its many advantages, HPLC does have some limitations that need to be considered in clinical diagnostics. Sample preparation. HPLC often requires extensive sample preparation, such as extraction, concentration, or derivatization, which can be time-consuming and labor-intensive. Cost. HPLC instrumentation, consumables, and maintenance can be expensive, which may limit its accessibilities for some laboratories or clinics. Skill requirement. Operating an HPL system and interpreting the results requires skilled personnel with specialized training, making it less suitable for point-of-care testing. Matrix effects. Complex biological samples such as blood or urine can cause matrix effects, which can interfere with the analysis and require additional sample cleanup or method optimization. In conclusion, HPLC, with its high resolution, sensitivity, and specificity, is a key tool in clinical diagnostics, with ongoing advancements promising to enhance its performance and extend its applications in biomarker detection and quantification. Thank you for watching.